I am Dr. Khokon Kumar Dutta. My Christian name is Dr. David Boone. I'm working as assistant professor in the Department of Biochemistry and Molecular Biology in Bangabandhu Sheikh Mujibur Rahman Science and Technology University in Gopalganj, Bangladesh. The title of my discussion is Discovery of mature microRNA-34a inside the cellular nucleus and the importances of nuclear microRNAs. I need to mention first that I was lucky to get the opportunity to pursue my PhD research in the School of Medicine, Kyoto University, Japan. One of my research projects was with microRNA-34. Some other researchers were also involved in this project. The environment of our laboratory was a little bit like a team of researchers. All the members of our laboratory tried to help one another as much as possible. I had co-workers in the laboratory from different other countries, but our senior doctoral researchers in the laboratory was the best help for me. He was a Japanese student. Interestingly, we were awarded PhD degree in the same year. He is now working as a faculty in the Nagoya University in Japan. A part of my project was to localize and to visualize microRNA-34a inside the cells. It was a very difficult attempt in that time. Nobody tried to do this type of experiments in mouse, rats, or even in humans around the world. Mature microRNA-34a, microRNA-34b, and microRNA-34c are very small RNA molecules. They had a very little differences in their sequences, which made them difficult to localize and to visualize separately. But we received great help from our Danish company, Khokon. We could collect a very specific probe, mercury, for the in situ detection and visualization of mature microRNA-34a. The probes are very expensive. I could collect that very easily because of the deep interest of my supervisor in the project. We prepared the rat tissues in the laboratory ourselves. We collected human tissue samples from South Korea. They were also very expensive. All the arrangements for a single experiment were economically so huge that the failure in only one time could be a matter of sadness, at least for me. But the Japanese usually need not to worry about money, at least in good purposes. The Danish company Khokon made those detection probes depending on the research published on lower animals. They could make microRNAs visualized grossly in the whole body. But in the experiments, we succeeded to localize and to visualize microRNA-34a in the cellular level and even in the different compartments inside the cells. We had special facilities and places for unrelated research in the department, we succeeded to visualize microRNA-34a in almost all cells in different organs of rat. Nobody actually tried this before. It was known at that point that the expression of microRNAs can vary in different organs. Something interesting got cleared from my experiment, that the expression of a single microRNA can vary among different cells even in a single organ. The expression of a single microRNA can even differ in different developmental stages of a single cell. At that time, everybody knew that mature microRNAs are produced and work in the cytoplasm of a cell. Even today in 2020, the same thing can be found sometimes in the textbook. I myself also knew the same information. But unexpectedly, we found the presence of mature microRNA-34a inside the cellular nucleus in my experiment. We did not have least idea about the functions and importances of mature microRNA inside the cellular nucleus. We only reported our observations in the research article. People usually get afraid of in case of saying something new out of the traditional thinking and belief. Our researcher strongly needs to explore the influences of experimental artifact and error in the first hand. It is essentially needed for a researcher to be confident and accurate about the experimental procedures and practices. Otherwise, a very little wrong information in any published article can cause great harm to our researcher and our research group. In 2019, I was trying to write a summary of my previous research in LinkedIn. I tried to check the details about the nuclear microRNAs and found that Thomas Sossel and his group in the USA discovered and published the presence of mature microRNA 21 in the nuclear fraction in July 2004, long before my unexpected discovery of microRNA-34a inside the nucleus. We were not informed about their big findings in this field. The group in the US who discovered for the first time the nuclear microRNA 21 were very famous for their works on small RNAs at that time. I myself was also used to keep my eyes on their works in a regular manner and try to take benefits from their published works. At that time, the concept of cytoplasm-centered functionality of microRNAs was so dominant in my mind, which led me to miss this great information. We actually could not use any supporting reference for nuclear microRNA-34a in my article. The group in the USA fractionated different cellular compartments and succeeded to detect signals for the presence of mature microRNAs in the nuclear fraction using northern blotting. But we succeeded to localize and to visualize mature microRNA-34a inside an intact nucleus using in situ hybridization approach. My supervisor was a famous pathologist doctor in Japan. He had huge expertise and knowledge in histology. He had precious research experience of working in the Food and Drug Administration, FDA, in the USA also. It is to mention here that he was honored with National Award of Japan in 2015 for his contribution in the pathology section of medical sciences. I think that he was satisfied with my experimental performances in his laboratory, and for certain he could realize the long-term impacts of the unexpected findings, which may promoted the inclusion of this observation in the article. Because he usually did the final modification and edit of a manuscript for submission in the scientific journal, a supervisor usually wants to be sure about the reproducibility of at least unexpected discovery using the same parameters and setup of the experiment. Because we see a PhD supervisor to be famous in scientific world, at least in the direction of his or her research. A famous scientist usually try to make sure the accuracy and authenticity of the contents of a research article before permitting someone to add his or her name in the author list. Anyway, our experiment may be considered as the first attempt globally, but today we know that a huge number of mature microRNAs work inside the cellular nucleus. It makes me a little bit happy because I invested a huge efforts for the in situ detection of microRNAs in Japan. The connections of nuclear microRNAs in normal physiology and in different diseases has already been explored to a notable level today. For example, cancer, hypoxia, metastasis, cancer-related angiogenesis, regulation of cytokine synthesis, inflammation, asthma, autoimmune disorders, and others. The known functions of mature microRNAs inside the nucleus include post-transcriptional gene silencing, transcriptional gene silencing, transcriptional gene activation, modulation of epigenetic mechanisms of gene regulation, and others. The translocation and transportation mechanisms of mature microRNAs from cytoplasm to nucleus has also been explored to some degrees today. Anyway, I would like to wish happy, prosperous, and dignified life for you everybody. Thank you.